So now I want to talk about a few things that happen after transcription initiation, which we looked at in great detail, at least the regulation of transcription initiation. And then we have the elongation step where you have the RNA polymerase and all the other factors that would be bound to it, moving down the template, transcribing RNA. And then at one point, it has to know when to stop transcription. So there are ways that signal transcription termination. And then in addition, the RNA that's made needs to be processed. That is, it's not final, the transcript is not ready to be translated into protein. And we're going to talk about two of those steps. One is 5' cap, which is at the very 5' end of the message. It gets modified. And then what's called polyadenylation at the 3' end of the transcript, where there's a bunch of adenines that are added. And then later there's splicing, but that'll take a whole lecture on its own. So this will be a kind of a shorter piece just to talk about a couple of these modifications that happen at the end of the transcription process. So here we have a very simplified drawing of the kind of the gene region, where we have the transcribe region in the open rectangular box, and then the transcription initiation site, the arrow, the promoter upstream, and then towards the end of the transcribe region there are termination signals that basically indicate that the RNA polymerase needs to stop, that we don't go on forever. And then you end up getting the transcript, which is the RNA molecule, 5' to 3', and that needs to be modified not in bacteria. It's modified only in the eukaryotes. And we'll talk about those modifications that take place in a little bit. But I want to start out just talking about the E. coli, because again, it's simpler. It's a little bit more well understood. And there is a particular sequence that stops transcription. The polymerase is released, the messenger RNA comes off the template. And interestingly, the actual sequence, I mean, it's encoded in the DNA, it's present in the DNA, so it's some information in the DNA. But the signal is actually carried by the RNA molecule that is transcribed. So I'll explain what happens there, at least some of what happens. So, of course, so here we're looking at E. coli termination sequence, and what you'll notice is that I'm showing you RNA. And you know it's RNA because you see U's in there. If it was DNA, there would be T's. And you have the 5' end, which is way upstream, and then we're really just looking only at 3' end, which is very close to the 3' end. Now, we look at the RNA sequence, what we see, or what you can figure out, is that there is internal homology. Just like we showed before, where you can have internal base pairing in a single-stranded DNA, you can also have internal base pairing in single-stranded RNA. And so if you look at this sequence, you can get this formation of these base pairs, which, if you look, are GC rich. And at the top, there's a loop, which is U, U, C, C, G. And then the base pairing is GC, AU, CG, CG, GC. Lots of G's and C's, and that's one of the important parts of this signal. So this is a GC-rich stem loop, and that's followed by A's and U's, and then it's the 3' end. So what happens is, relatively, in a way, straightforward, if you look at the bottom, it's showing basically the DNA opened up through a helicase. The RNA polymerase has come along, has made the RNA molecule. Once it passes the DNA sequence that gives rise to that sequence that can form a stem loop, it immediately forms a stem loop structure, like instantaneously. And then you have all the A's and U's. And what happens is, so the stem loop structure binds a protein called NUS-A. So it's recognized I think I told you a couple weeks ago or a couple lectures ago that these stem loop structures are often used to bind proteins specifically. And so that's what's happening here. You have the specific stem loop that's synthesized, the NUS-A binds to that, and the NUS-A inhibits the RNA polymerase from continuing transcription, and then also destabilizes it from the template. So it comes off. And the reason it comes off rather easily is because there are just a whole series of A's and U's, and those base pairs are a lot weaker, so that it falls off. So the two important points, or three important points, is you need to have the GC-rich stem loop. It does not require a specific sequence as long as it's GC-rich. So you need the GC-rich stem loop, you need the run of A's and U's at the 3' end for the unstable base pairing, and then you need the NUS-A to bind to inhibit and destabilize the polymerase, and then the template comes off, the RNA comes off the template, and the RNA polymerase comes off the template, and so that's termination. Now, in bacteria, E. coli. Now, in eukaryotic cells, it's not quite like that. It's actually a little bit more complicated. It's a little strange. I'll show you what we think happens here. So this is eukaryotic termination. Now you have a messenger RNA with a 5' end and 3' end. Actually, this is a primary transcript because it still needs to be modified. But if you look at the end of the message, near the 3' end, 10 to 20 nucleotides from the 3' end is the sequence AAU AAA. It's pretty invariable. AAU AAA. That sequence is required for two things to happen. It's required for proper termination of the transcript, and it's required for polyadenylation, which we'll talk about in a few minutes. So it's not, okay, so I'll show you, it's not termination per se, but what happens is the RNA gets cut 10 to 20 nucleotides downstream from that sequence. So it shows below. So you have the transcription going with the RNA polymerase II with the elongation complex. And so there's a bunch of proteins that are bound to it. Of course, the helicase is there. It's not shown, but there's these other proteins. These are the ones that seem to be involved in the cutting process. So what happens is after the polymerase passes that AAU-AAA sequence, or synthesize that sequence, there's an activation of those three proteins that cuts the transcript. It cuts the transcript. So there's CF1, which stands for cleavage factor 1 and cleavage factor 2. And then there's cleavage and polyadenylation specificity factor also there. So these proteins cut the end of the message, generates the proper 3' end, but interestingly, and that's what happens is this polymerase keeps going. So it continues transcribing. So it's not termination per se, it's just that it ends the transcript with the cut. The polymerase continues to go it can go for another 1,000 or 2,000 nucleotides, and then it'll just eventually fall off, and probably what happens is some of these elongation factors and other factors that are part of this complex just start losing their affinity and then leave the polymerase, and it stops. So we have termination in E. coli, and we have eukaryotic quote-unquote termination. Really, it's generating the 3' end by a cutting reaction. So these transcripts, as presented, particularly this one here with the 5' and 3' end, is not capable to be used as a messenger RNA, because the messenger RNAs the transcripts need to undergo a modification, a few modifications that take place to make it a mature message. So here we show again the open rectangle is the transcribed unit, plus one site is the initiation site, the Tata box, and so on, and you get transcription. And those dark boxes represent the exons, which have the coding information for the protein, the proper codons to make the proper protein, and then the white boxes are the introns that will be removed. So the first thing that happens, you get transcription, and we have a primary transcript which is capped at the 5' end, and it's polyadenylated. So those are two modifications that take place we're going to look at. And then the splicing, which we'll look at next time, is where you have the introns removed and the exons joined together. So first let's talk about the 5' cap and how it's added and what it's used for. So the 5' cap, added by a specific enzyme, which is called guanyl transferase. It's actually part of the elongation complex, which is carried along with the RNA polymerase too. It actually happens very quickly. The way, a lot of times the way they have it drawn in the textbook, or even sometimes the way I draw it here, you have the primary transcript, then they show it being modified with a 5' cap. But that's really not the way it happens. The 5' cap is added very soon after initiation. In fact, after around 30 nucleotides are synthesized, the cap is already on there. So it happens very soon, because it's part of the elongation complex. So it's added by the guanyl transferase. Let's take a look at a drawing of what it looks like. So if you look on the right-hand side, that is a short stretch of RNA. The very first one at the top that has A or G, it's usually the beginning, the first transcript, the first nucleotide transcribed is either A or G, not always, but often. That's the +1 nucleotide, meaning that's the first one that's made by the polymerase. And then the next one down is +2, which is a uracil. Next one down, I have a question mark because it doesn't matter what it is. First thing I want you to notice is that the first two nucleotides have a CH3 added to the 2' hydroxyl. So usually, if you look at the third one with the question mark, there's the 2' hydroxyl, which we talked about as what is the ribose and what makes the RNA active, reactive, and then but the first two they're methylated. So those two become non-reactive, and it's thought to stabilize the 5' end of the message. And then the cap is shown up at the top towards the left. The 5' end is that CH2 with the 3 phosphates. And then the cap that's added is this methylated guanyl residue. It's a RNA nucleotide because it has 2 OHs. The guanine is on there, but it has an additional methyl group added to it. It's a little bit modified, and it's attached to the triphosphate by the 5' carbon. So this is a little bit of an unusual linkage. It's a 5' to 5' triphosphate linkage. That is the cap. It's methylated guanine attached to the 5' trimethylated triphospho linkage to the 5' end of the first nucleotide. What's the point of this? Well, as far as we can tell, it's essential for ribosome recognition. That is, there's lots of RNAs in the cytoplasm, lots, and there's a lot more than we ever recognized. All kinds of short, small RNAs and long non-coding RNAs, things that don't encode protein. And so the way the ribosome recognizes specifically that there's a messenger RNA is that there's the 5' cap. So there's a receptor on the ribosome that binds to the 5' cap and pulls it in. And then it also is required for stability of the messenger RNA. There's nucleases in the cytoplasm and the nucleus that will degrade and chew off nucleotides from the start, also from the 3' end, also in the middle, but this helps stabilize the message. So that's the 5' cap. And then the last thing I want to talk about today is the polyadenylation. So the 3' end of all messenger RNAs except histone messenger RNAs, every other messenger RNA in the cell is polyadenylated. Polyadenylation is a series of adenine residues that's at the 3' end. So what's required is the AAU-AAA sequence that activates the enzyme that causes the polyadenylation. It's added by an enzyme called polyA polymerase. That's all this enzyme does. It doesn't transcribe RNA. It makes RNA because it's making a long run of adenines. So it is making RNA, but it doesn't transcribe like a RNA polymerase 2. And the other thing is that this RNA polymerase, this polyA polymerase, does not require a template. All it does is it binds to the 3' end of the message and then just synthesizes and ligates together, or polymerizes a whole series of adenines that can be anywhere from 50 to more than 300. So it's a long, what we call poly-A tail. The functions of this are a number. First of all, it's required for proper export from the nucleus because it binds, the poly-A tail binds to a protein called poly-A binding protein. This participates in getting it out of the nucleus because these are messenger RNAs that need to be translated. So helps get it out through the nuclear pore, through into the cytoplasm. It's also required for proper splicing. It participates in the splicing reaction that we'll talk about next time. It helps stabilize the messenger RNA. And also it participates in efficient translation. So it's required for all these things. Now the poly-A tail is also used as a tool to purify messenger RNA. A lot of times when we see certain properties of molecules, we can use that to our advantage to manipulate them, or in this case, purify. So if you're interested in looking at a messenger RNA, studying what does a messenger RNA look like, what are some of its properties, it would be hard to do because the messenger RNA is a minority of RNA in the cell. If you look at the total amount of RNA in the cell, 75% of it is ribosomal RNA. 20% of it is transfer RNA. Around 3% is non-coding. Some of these long non-coding RNAs, also the microRNAs that I talked a little bit about, I'll talk more later. Messenger RNA is only 2%. So 98% of the RNA is something else. So if you're interested in studying messenger RNA, it's going to be swamped out by everything else that's in there. So we take advantage of the poly-A tail to purify the RNA from the mixture. And it's based on the ability of these long stretches of A's to base pair with long stretches of T's. Normal base pairing. So we use what's called oligo-DT affinity chromatography. This is similar principle to the chromatography that we used a couple days ago to purify the DNA binding proteins. It's very analogous, but it's oligo-DT. Oligo-DT means it's an oligomer, a length of thymidine residues, basically a DNA run of T's. So we take this run of T's, you can synthesize these, you can purchase them, and you link them again to these small plastic resin beads, very tiny beads, and I've amplified or magnified one out, and so you can see a bead attached at the 3' end to the bead. So the 5' end is out, hanging out in solution, and these beads can have many of these attached to it, so it's not just one. And then you take these columns, which is a spout on the bottom, and you pack it with this resin. So what you've done is you're basically packing it with immobilized oligo-DT sequences. And then what you do, and the notion is that the poly-A tail will base pair to the T's. So you take total RNA, total cell RNA. The goal is to purify messenger RNA. You take total cell RNA, put it onto the column, and let it run through. And you wash that column with saline buffer. And so on the bottom right, I have wash. So as you're washing it, RNA comes out. Lots of RNA comes out. All the ribosomal RNA, the transfer RNA, all the other RNA comes out because it doesn't have polyA. So everything comes out, and you can monitor that, and you can see a lot of RNA coming out, and then eventually you see no RNA coming out. At that point, you switch the buffer, and you can add a mild detergent, which will break the base pairs because they're not real strong base pairs, or you can increase the heat, to, even probably 50, 55 degrees, which will cause the A's and T's to come apart because there are only two base pairs, they're not as strong. And then what happens is some RNA comes out, and when you analyze that, it's pure polyA plus messenger RNA. So the polyA tail is used by the cell, and in this case it's also used by us to purify pure messenger RNA from a mixture. Okay, that does it for today. Have a good day.